So diagnosis, how do you diagnose the case of phenylketonuria? So since the block is there, so phenylalanine level in the blood will be very high in the blood. So first and foremost, you can estimate that phenylalanine in the blood, or basically if you want to diagnose the disease before birth, while in the womb, you can make a prenatal diagnosis by taking a chorionic villus sampling or from maternal blood plasma, taking the DNA of that particular baby, which will be available. As I told you earlier, you can do this DNA test, so that a mutation you can have to detect. So nowadays it is possible because of the next generation sequencing is available, or PCR based test. Tandem Mass Spectrometry is the most standard method and most widely adopted method for any inborn errors of metabolism disorder. So blood phenylalanine by this tandem mass spectrometry is not available everywhere. I mean, this is also costly. Neonatal diagnosis by measuring phenylalanine level. It is very difficult to get in which laboratory it is being done regularly, daily. These are all done by specialized laboratory. It's not that easy. So its level, there will be, because of the breakdown, wear and tear, some amount of everything will be present in the blood. And the normal amount of phenylalanine will be about 1 milligram per deciliter. Now, since there is a phenylalaninemia, so that level will be high. So there must be a cutoff limit. What should be the cutoff limit for disease detection? Usually cutoff limit here it is taken to be 30 milligram per deciliter. But even if the value hovers somewhere around that, and if there is another test positive, you can make a diagnosis of phenylalanine hydroxylase deficiency. So estimation of phenyl hydroxylase enzyme itself, it is a very difficult question. It is where it is done. We talk too much things in the theory, but in reality, if you want to do that test, it is not done. It is not done here, it is not done there, it is not done that. A Guthrie's test, which I will be telling you, it's a rapid screening test, and then ferric chloride test is a crude test, it is not a specific test. So Guthrie's test I described, in the year 1988 to 1991 in 4 MBBS batches in a practical class, using how it is done with the bacteria petri dish. Since it is a theory class, I will try to simulate as much as possible. So sample collection of blood from the adult can be done from vein, but from a newborn baby it is very difficult, or 2 to 3 year old baby. So there is a standard technique, that is heel prick. So this is how it is done. The heel will be held like this, then there will be a puncture will be made, and the blood will come out, and blood will be collected on the filled paper strips in a circular spot. These are the spots, the white circle. Ultimately, these type of blood-soaked filter paper will be sent to the laboratory for analysis. That is how neonatal blood testing or screening is done. Usually a few drops of blood are obtained from a small prick made on the heel, and it allows early identification of any biochemical parameter as well as genetic testing. Because blood is available, DNA will be available, you can do the test. So tandem mass spectrometry, as I told you, is the most preferred method. So this is not available, but if you suspect, you can send it. A battery of tests of 40 tests, different tests can be done, maybe within an hour or so. So that test, many child health institutes or pediatric care or a pediatric department, they are using it for clinical purposes as well as for research purposes. Now, Guthrie's test is called Guthrie Bacterial Inhibition Assay. Now, what is done? I am trying to say. See, this is. Can you see this structure? There is a handle, there is a wire, and on the top there is a circular thing. Usually, what is done? These are bacterial culture dishes, plates. This I could have shown it physically to you in a practical class only. That's what I have brought in, that I've been almost 40 years, nearly 35 years back. So this is the plate actually. Here some materials are given so that bacteria can grow. Bacteria needs food. All bacteria need food. There is no doubt. These are the food ingredients which are given in the bacterial plate so that bacteria can grow. Now, if suppose this is an urine sample, if there is an infection in the urine, so bacteria will be present. So, if you do not make it sterile, then that particular loop will contain some bacteria and that will contaminate. So, to prevent that, it is made red hot. It is made red hot in a flame. Then it is cooled. Then it is inserted into the urine so that only thin film of urine is present in the small circular hole. That is rubbed. That is taken in the culture plate and that is rubbed here and there. Now, if there is no bacteria in the urine, no infection, the culture plate will remain like this. If there is a bacteria, then bacteria will start growing. The more the bacteria, more the growth. The less the bacteria, less the growth. You can see a variety of growth will be available. Now, this test is based on inhibition assay. So, here what is done, in this media, one inhibitor is added. So, Bacillus subtilis is the bacteria which is chosen. The inhibitor for bacterial subtilis growth is beta-2 phenylalanine. That is added so that even if the bacteria is present, bacteria will not be able to grow because it prevents the growth of Bacillus subtilis. Now, we have collected blood in the filter paper. And suppose the baby is having phenylketonuria or phenylalanine level will be high. So, that amount of phenylalanine will competitively inhibit with the beta-2 alanine, the beta-2 phenylalanine, and then it will percolate into the medium because they require Bacillus subtilis will require phenylalanine to grow. So, there will be growth around the petri dishes in the culture like this here. You see positive. You see this circular spot I am telling you, the black one is the filtered paper strip which had high phenylalanine level. It has overcome the action of beta-2 phenylalanine and it spreaded into the medium, and that's why there is a bacterial growth. Okay, so that is what is bacterial inhibition. This is negative, there is no growth, and this is positive, there is a positive control, means there is a bacterial growth. So this is negative control. So this is how this test is done. The ferric chloride test is another test because the ketone group is there. So ferric chloride test will be positive, and you will do ferric chloride test in your practical class. If phenylpyruvate basically is having a ketone group, that is the reason ferric chloride test will be positive. This you will see in your practical class, and for ketone body also you will do a Rothera test. So Rothera test might also be positive because of the phenylketone. So the incidence what is told, it is in 18,300. Today in the morning during breakfast, I asked one of my senior batchmate of 3 years. He is right about 67 years. He is a pediatrician. He has seen throughout the cases, I mean a large number of cases, but he had not seen a single case of phenylketonuria in his lifetime. And nobody. I have inquired from many persons. So it is the most common inborn errors of metabolism. It is said, but have you found a case anywhere? No. Answer is no. So all these figures of incidences, whatever you will read, are not real figures. They have been copied somewhere from somewhere and pasted it. And in India there is no data. Actually there is no data. So whatever way you say, these data are not acceptable. So you will find the data, but the data are not real. So precaution what you have to take. So individual must not take anything, any substance which will contain phenylalanine, then that will give a false positive result. So there will be some person, some woman who wants to be a mother, so they are to be kept on in a childbearing age, planning for a pregnancy. They should be kept on a low phenylalanine diet before conception and throughout pregnancy. That is a mandatory requirement because there is a risk of miscarriage, and particularly no woman want to have a baby with a defect. So treatment again, there is no cure, no treatment. No treatment basically. Treatment is there, there is no cure, right? Treatment is there is no cure. Only once the baby is detected to be phenylalanine, some damage has already been taken place before the diagnosis is made. Now once the disease has been diagnosed, the phenylalanine free diet is to be given, number one. Now to get a phenylalanine free diet is very difficult. A newborn baby will need mother's milk. Now if mother milk is taken, the phenylalanine will be there. So what we have to do to prevent, allow a phenylalanine load to be as minimum as possible, the baby has to be taken out from the breast frequently, which is very difficult to practice. Baby wants to suck the milk from the mother because of the hunger. You cannot take the baby away from the mother's breast. It is difficult to implement, but we have to advise in this case. And then we have to devise some low phenylalanine diet for those type of babies, particularly up to the age of 14 years, so that the high ill effects of phenylalanine on growth and will not be there. So since phenylalanine is an essential amino acid, several other body protein synthesis will be required. So we have to give the diet with phenylalanine, but it will be of low phenylalanine containing diet. So that the special diet will be required, normal diet will not be able to provide that thing. And in the mother while a breastfed baby is there, only control mechanism is to allow breastfeeding at a frequent intervals and not large amount at a time. What normally babies are taking, that is the only advice you can give. So after the age of 14 years, when the growth will almost complete, then you can give, there will be not much problem. So Jambulkar is very favorite book, I know. 3 days back, Jambulkar made a WhatsApp status. I mean, I got a screenshot made from Jambulkar. His number is with me. I occasionally contact. So phenylketonuria for NEET-PG examination. So it is a favorite question for your UG entrance of this final MBBS examination, this first MBBS passport examination. So catabolic pathway of phenylalanine, I told you about the first step. There are other areas. The two are reversible reactions, and 3 types of tyrosinemia are there. One is the last step is the first one, that is tyrosinemia 1, about which I will tell you. This is Tyrosinemia 2 here, Tyrosinemia 3 here, and Alkaptonuria is another part. So cleavage of aromatic ring is done by the homogenous acid and to malonyl acetoacetate. So homogenous 1,2-dioxygenase is another enzyme which can be blocked, and that will lead to accumulation of homogenous. So what will happen here? You see, homogenous will get actually oxidized to polyphenol oxidase, and alkapton bodies will be formed. So they will cause such type of black deposition in the intravertebral disc, and in the eyes you can see this is ochronosis. This is called ochronosis. Once you see this type of cases, then you will be able to think, diagnose that this can be case of alkapton body, and this can be deposited on the cartilage of the bone, and urine, fresh urine will not look abnormal, but on standing it will form a black urine. That is called alkaptonuria. Okay. So polymerization of this homogenous set will cause alkapton bodies, and these will be deposited in the eyes, nose, ear, and the intravertebral disc spaces. So you see there will be a block here, homogenous 1,2-dioxygenase, and you will get an urine color. It will be on standing, it will be oxidized to give a black color, which is a common presentation. Now normally we pass urine, don't bother for what is the color after some time. That is an usual habit, and that happens because of this thing. The disease remains unnoticed, but usually in the underwear of the baby, it is not removed. There might be blackening, and that might arouse a suspicion to the mother that the nappy is not normal, but it is having a black spot because it is getting an exposure to oxygen. Okay, but if the urine is passed through, then this disease cannot be detected. So the earlier the disease can be detected, earlier treatment can be instituted, earlier diagnosis can be made. Do you get my point? So again, 2 days back again Alkaptonuria was there, so every day I have been putting an update. So another is Tyrosinemia 2. The block is here, tyrosine aminotransferase. It is basically allowing tyrosine to para-hydroxyphenylpyruvate. This stage is blocked. So this causes oculocutaneous tyrosinemia. So Tyrosinemia 2, there is a block in the tyrosine aminotransferase. This catalyzes the reaction from tyrosine to para-hydroxyphenylpyruvate, and that causes oculocutaneous tyrosinemia. You see oculocutaneous tyrosinemia. This is the appearance. So whenever you examine a body, examine thoroughly so that you don't miss anything. Who cares for the legs? Nobody. But you see here Dr. Nida Brahmanik, I have seen, I will sit every Saturday with him. Any patient's leg, he takes and keeps on his lap to examine thoroughly what is there under the sole. Which doctor will do that thing? I am quite amazed to see. Even, patient feels embarrassed that a doctor is taking his leg back, right on his lap, and then he is examining. This much passionate doctor he is. So I have seen the under surface of the foot by virtue of him, otherwise I could not also have seen such type of patients. So you will not miss anything. Eyes, you can see, if you look at the eyes, for all patients you will be able to diagnose one day. So another copper containing enzyme is phenyl hydroxypyruvate. The previous stage to next stage is homogenous. That is Tyrosinemia 3. So last stage is important. One says that at the end, basically, this fumarate acetoacetatase or fumarate acetoacetate hydrolase basically breaks down fumarate acetoacetate to fumarate and acetoacetyl-CoA. Now, if this enzyme is blocked, you see, because it yields finally to two structures, one is fumarate, which is glucogenic, and acetoacetyl-CoA, which is ketogenic. Phenylalanine acts both as, and tyrosine both acts as glucogenic and ketogenic amino acid. Now, if this stage is blocked, you see, if this stage is blocked, fumarate acetate, this enzyme is blocked, so fumarate acetate will be accumulated, isn't it? If this enzyme is absent, so there will be no breakdown of fumarate acetate to fumarate and acetoacetate. So fumarate acetoacetate will be accumulated, and you see what will happen. See, fumarate acetoacetate will be there, it will be converted to succinyl acetoacetate, and this succinyl acetoacetate will inhibit heme synthesis. Heme production will be compromised. So since heme production will be compromised, hemoglobin will not be formed. It will be formed in low amount. So anemia will be there. Okay. So fumarate acetoacetate or fumarate acetoacetate hydroxylase will cause Tyrosinemia type 1 type, and it will be diagnosed by high succinyl acetone level, because this fumarate acetoacetate will be converted to succinyl acetoacetate, and this is basically measured in the blood, and that will inhibit heme synthesis. So because of the high concentration, there will be cabbage-like odor of the urine. So there will be feces will be cabbage-like, urine will be there. So cabbage-like odor is specific for Tyrosinemia type 1. This you note down as a short note, as an MCQ or high succinyl acetone level. These are the two things which I want to tell you. This metabolism will get in detail some of the points which you note down, because this straight cut question will not come, question will come in a very tilted way, and that you have to answer in your own ways. These are the areas which I will highlight. Okay, with the pathway you have to remember where the block is there, and a typical question I will tell you what type of question will be asked, because nowadays questions are not that straight. Even if you read thoroughly, you will not be able to answer. So first and foremost, tryptophan is an 11 carbon atom. It is fully catabolized into 11 carbon atoms. How? I will show it to you. So this is, you remember, this is what type of ring it is? What type of ring it is? Aromatic, benzene, indole, imidazole. Indole, yes, indole. Correct. Because you see, this is the structure of benzene, this is the structure of imidazole. So this is a combination. Correct. So this is the pathway. So I have written here 3-hydroxyanthranilic acid, and this is 3-hydroxyanthranilic acid. I have brought it here so that you can see in one cycle. So the reason why I have shown it to you, there are 3 carbon dioxide. You see, the 1 carbon dioxide is formed here, 2 carbon dioxide is formed here. So 3 carbon dioxide is catabolized as CO2. There are 11 carbon. This slide is basically to show you the release of carbon dioxide. And several other things are there. I will discuss each and every point. See here, end product, one is alanine, one is acetoacetate. At the end, what we are getting? We are getting acetoacetyl and we are getting alanine. You see, when the tryptophan is broken down at one point of time in this reaction, we are getting alanine out from tryptophan. Okay? So there is an alanine is an amino acid and acetoacetate is another amino acid. So at the end, what we are getting? 3 carbon dioxide I have already shown. So 3 carbon is out. Now acetoacetic acid was there. We have seen at the end, this is having 4 carbon and formyl group is there. Alanine is here. This 1 carbon unit is formyl carbon unit, one unit here. Alanine is having 3 carbon unit and acetoacetate is having 3 carbon unit. Okay? 4 carbon unit acetoacetic acid and alanine 3. So it will make total 11. So this is the major end products. You can see from tryptophan how many useful amino materials are coming. One is serotonin, one is melatonin, one is 5-hydroxyindole acetic acid and indical. These 2 are excretory products. And we are getting serotonin, we are getting melatonin, we are getting alanine, we are getting acetoacetyl-CoA, and we are getting formyl methyl tetrahydrofolate. In addition, tryptophan is also participating in the protein synthesis also. So useful products from tryptophan are basically formyl 1 carbon unit, serotonin, melatonin, alanine, acetoacetate. Okay? And NAD. NAD is another important thing. NAD we are getting, and NADP also we can get. So biologically important substance produced from tryptophan is alanine. Alanine is a glucogenic amino acid. Acetoacetyl-CoA, as I told you, it is a ketogenic. Formyl group 1 carbon unit, it participates, I told you. Niacin we get. And this is the area where I will discuss a little bit because tricky questions will be asked from this part. Simplest question, what are the biological important substances which are produced from tryptophan? And the answers are 6. Alanine, acetoacetyl-CoA, formyl group, niacin, serotonin, melatonin. And excretory product, as I told you, hydroxyindole acetic acid and indican. So 3 pathways are there. This is major catabolic pathway is the formyl kynurenine ancylate. And here 3 carbon dioxide is produced. I have shown it to you. And there is another pathway from here in the middle. The pathway is breaking and forming niacin, nicotinamide mononucleotide, nicotinamide dinucleotide. Serotonin and melatonin synthesis pathway, there is another pathway, but it is from in the middle of this pathway. So ideally there are 2 pathways. 3 pathways I have delivered written so that you remember. One is major catabolic pathway, that is a standard kynurenine ancylate. Why? Because these kynurenine and ancylate are the intermediate products of the this tryptophan metabolism. That's why this is called a kynurenine ancylate pathway, and that contributes to nearly 90, some books write 95%, some 95.7%. So more than 95% are catabolized in that pathway, and only 1% of the pathway or 3% of the pathway takes part in the nicotinic acid pathway. And of course, still some lower part is for the serotonin and melatonin because we require in lesser amount. So you see why it is called kynurenine and ancylate. So kynurenine is produced here, you see in this reaction. And ancylate is there, this 3-hydroxy ancylate acid. This is the reason it is called a kynurenine pathway or kynurenine ancylate pathway. So this is the major pathway of tryptophan metabolism. Okay, so here this step you remember here. So from tryptophan to N-formyl kynurenine to kynurenine, and in this stage, this 1 carbon pool, that is formyl tetrahydrofolate, is formed here. And from kynurenine to 3-hydroxykynurenine, this step is important. I will tell you about this step where it is related to plaque and niacin deficiency. So, this 3-hydroxyanthranilic acid is the part from where the main route is going, and other 2 routes are also from 3-hydroxyanthranilic acid also, that is NAD or niacin or your nicotinamide mononucleotide, dinucleotide, or this melatonin serotonin pathway. Okay. So, you see here this same structure what I have shown it to you. So, this 1 carbon unit is produced from this 1 carbon unit pool is formed from the phenylketonuria pathway. Okay, that's why I have written this 1 carbon unit is coming from this pathway. The normal pathway is this one, 3-hydroxyanthranilic acid to this one, acid-to-acetyl-CoA. You see the major pathway, acid-to-acetyl-CoA is the end product here. So, this is the major pathway is producing acid-to-acetyl-CoA, and now from 3-hydroxyanthranilic acid, there is one pathway which is producing niacin and that is a vitamin or nicotinic acid, and on niacin mononucleotide, and from niacin mononucleotide to dinucleotide. Okay. So, QRPT is the, you see note down here, this you note down as the rate limiting step. So, MCQ is likely from this part, this question. The QRPT enzyme is the rate limiting step of tryptophan metabolism. These are the points you note down, rest you just read it from the book. I mean, this is also written, these are not written, but highlighting points. So, synthesis of serotonin is from tryptophan through 5-hydroxytryptamine. 5-hydroxy first is decarboxylation, first is tetrahydrobiopterin is required here. It is an analogous reaction just like phenylalanine hydroxylase. So, tryptophan hydroxylase enzyme is there. Here again tetrahydrobiopterin is required. It is analogous to phenylalanine hydroxylase enzyme, where the tetrahydrobiopterin needs to be regenerated through NADPH. It is the same pathway. Just remember that one, there oxygen was required, water was produced, here oxygen is required, also water is produced. Now, from here hydroxylation reaction, and that is a decarboxylation reaction. So, this decarboxylation reaction produces 5-hydroxytryptamine, and that is basically serotonin. 5-hydroxytryptamine is serotonin. So, first step is hydroxylation, second step is decarboxylation, and that is the production of serotonin. Now, from 5-hydroxytryptamine, that is serotonin, an excretory product can be formed, that is called a 5-hydroxyindole acetic acid, or it can produce further down to produce melatonin. Yes, adenosyl methionine, I told you and I will tell you in a short while again. Yes, adenosyl methionine is the active form of methionine, and that takes part in this reaction, and it transfers the methyl group, and then acetyl serotonin is converted into melatonin. So, from serotonin in a single chart, you will get melatonin, you will get 5-hydroxyindole acetic acid. So, serotonin and 5-hydroxyindole acetic acid are produced from melatonin, and 5-hydroxyindole acetic acid are produced from serotonin. See, this cycle I have been showing you in every reaction. I will show you to show which one is glucogenic, which one is ketogenic. Now, you see tryptophan. Tryptophan is, you see here, tryptophan is producing, here this blue one is basically ketogenic and red one is glucogenic. So, tryptophan is producing pyruvate, pyruvate is participating in glucogenic reaction, and tryptophan is producing ketone body, acetyl-CoA, acetyl-acetyl-CoA. So, why it is an ketogenic, why it is an glucogenic, that we can remember. Now this Kynurenine pathway I have shown it to you. Now I am telling you one more significance. You see here, kynurenine is produced from kynurenine to 3-hydroxyanthranilic acid. Pyridoxal phosphate is vitamin B6. Vitamin B6 is required. Now, tryptophan pyrrolase is the first reaction. This reaction is the first reaction is tryptophan pyrrolase. So, that is basically having an indole ring. It incorporates N-phenylketonuria. It is inducible by the liver. I mean, this reaction is note down here. This enzyme, kynureninase. This kynureninase enzyme requires pyridoxal phosphate. Pyridoxal deficiency will not allow this reaction to proceed. So, xanthinuric acid will be produced. So, if you look at the reaction, if you give a tryptophan load, tryptophan will come to this stage. But because of the kynurenine deficiency, secondary to vitamin B6, this will not be produced. So, xanthinuric acid will be produced from 3-hydroxykynurenine. So, that will be produced in response to a tryptophan load, and that will cause to niacin deficiency. So, if you don't give vitamin B6, there will be another deficiency of the vitamin. And because of the niacin deficiency causes pellagra, vitamin B6 deficiency can cause pellagra. Explain, justify why vitamin B6 can cause pellagra. Now, this reaction you have to tell how vitamin deficiency is occurring, because it is blocking the kynurenine enzyme. Okay. So, 60 milligram, again niacin is produced from tryptophan. Niacin is produced from tryptophan. So 60 milligram tryptophan will be consumed. Then only 1 milligram of niacin will be produced. Okay. But if the diet is deficient in tryptophan, then niacin will not be produced. If you don't take niacin from other sources, then there will be deficiency of niacin also. So, pellagra will also be there. So, those persons who are where the maize is staple, that maize is deficient in tryptophan. So, poor persons who are living exclusively on maize and there is no other food substances, so they can also have pellagra because they cannot produce niacin in adequate amount. So, this is the normal pathway, this is the synthesis of niacin, I have shown it to you. So, if there is a block here, PLP, that is vitamin B6, this kynurenine enzyme requires PLP to optimally function. So, if there is a block, the alanine will not be produced and xanthinuric acid will be produced in urine, and so there will be formation of xanthinurate in B6 deficiency. So, a question again can be asked, why xanthinuric acid is present in vitamin B6 deficiency? Explain, justify. These are the answers, these are the questions will be asked from tryptophan metabolism, not the straight cut question. Why xanthinuric acid? Xanthinuric acid is found, or xanthinuric acid is found in urine in vitamin B6 deficiency, or in case of kynurenine deficiency, when the kynurenine cannot work. Kynurenase, PLP, whatever way you see, this question will be asked. We want to know that this reaction is not occurring, conversion from 3-hydroxykynurenine to 3-hydroxyanthranilic acid. So, because of that thing, this is accumulated in the urine. That will be the answer. A synthesis of NAD to again NADP and NADP also. So from NAD can be produced, from NADP another phosphate will be added, and this NADP will be used also. So from niacin, if niacin is not produced in adequate amount, NAD will not be produced. And you know there are many reactions which are NAD dependent, NAD dependent. So nicotinic acid will be blocked, nicotinic acid NAD synthesis will be also compromised because block is here. So you see what is the importance of taking vitamin, and one more important will be discussion will be there in the next one. So synthesis of serotonin, as I have told you, serotonin catabolized to indole acetic acid, I have explained. Serotonin to melatonin synthesis, I have also told you. Only thing you have to remember. So 5-hydroxytryptamine, write a short note. You can write down this thing. It is a neurotransmitter. Its availability is basically it induces sleep. Protein-rich food. When you amino acid, tryptophan amino acid is a large amino acid. So when you take a huge amount of protein, then there will be too many amino acids. And since this is a bulkier amino acids, so there will be competition for absorption. So other amino acids will be absorbed in preference. So tryptophan absorption will take a longer time. So because of that thing, there is some amount of tryptophan deficiency temporarily within the cell. So that's what I want to say. This major exercise, this you read from the book. 3 diseases are related to tryptophan. One is heart nerve disease. Heart nerve disease I have told you in your amino acid class, in your protein metabolism absorption class, digestion and absorption. Only point I have told you that only one amino acid that is related to tryptophan is absorption is blocked. Now it is not so, not only tryptophan but other neutral amino acids. There is a common transporter. So other neutral amino acids will not also be absorbed from the intestine and also from the renal tubular cells. So there will be amino aciduria. So the concerned gene is SLC4. You may not remember, don't remember. If you cannot remember, doesn't matter. But this is a gene, the encoding protein is this one. This is a sodium dependent co-transporter. So a number of neutral amino acid will be excreted in the urine. So defective tryptophan absorption, and as I told you, maize is deficient in tryptophan. So niacin is present in maize, but it is attached to a protein called niacin, and we don't have in our body to break that bond from that maize. So that's why that is not absorbed. So that is one important aspect of maize. So maize eating population, if they don't consume niacin from other sources, they are also will have a pellagra-like symptom. So niacin biosynthesis, if it is impaired, it causes a pellagra, and pellagra is characterized by 3 Ds. One is diarrhea, one is dementia, one is dermatitis. 3 Ds of pellagra: diarrhea, dermatitis, dementia. That you remember. Daily requirement in adult is 12 to 20 milligram. So we have to take a good amount of niacin also. So pellagra patients look like this. Now if you get a blackish patch, 3 things I told you: diarrhea, dermatitis, dementia. Because disease will be the patient will not come overnight. They will observe, for the chronic patients will come and they will get this type of patches. And then you can diagnose, if it is a low socioeconomic background, have a suspicion in mind that this might be a case of pellagra. Second disease, tryptophan is synthesized by the argintophene cells of the intestine. So it is present in the intestinal mucosa, intestine, as well as a brain, but argentinophenoma is basically describing the tumors or paraneoplastic syndrome of the intestine. So these cells will undergo paraneoplastic changes and that will cause a carcinoid tumor. So normally 1% of tryptophan is channeled to produce serotonin, and major part of the serotonin in the body is produced by the gut. So when there is a tumor, 60% of the serotonin tryptophan will be converted into serotonin. So there will be temporary niacin deficiency and urine hydroxyindole acetic acid will be increased in high amount. So there will be symptoms like flushing and sweating, not sitting, sweating, intermittent diarrhea. Of course, melanin is a pigment which is present in the skin produced by the melanocyte. Melatonin is a hormone which is produced by the pineal gland. These are two different things. Melanin is produced from tyrosine. Melatonin is produced from tryptophan. So, xanthinolate in urine, I have told you why vitamin B deficiency it occurs. So, I am ending. Last point, I just take 10 minutes more and then I will end. Regina, part. Only highlights I will show and then I will end. Methionine to dekhe to arginine. Many things have been told in the previous class. It is a repetition of just recollection of what I have told you. I will add 1 or 2 slides more, not more than that, but previous recapitulation only. You will understand that all these things have been told in earlier. See, what is the difference? This slide I have shown you during an amino acid class. So, just to show sulfidyl group, here you see the sulfidyl group is in the free form, acid form. Now, it is in the form of thioester form. Now, in this form, this removal of sulfur will be very difficult. This is the reason why I have shown this structure deliberately during that time and this time also. Now, removal of sulfur, sulfur here it can be oxidized. In the glutathione you have seen GSH to SSG and to DSH. Because it is here free, but a thioester bond, it is in the thioester bond is very difficult. So, this has to be somehow activated. This sulfur is to be used. And again this metabolic fate of carbon skeleton, you can see it, methionine. All the amino acids, either glucogenic or neogenic, this same cycle, I will show everywhere. And you see, you will get an answer from the same cycle for all the amino acids. So, this is basically a glucogenic amino acid, it will enter as a production succinyl CoA. So, what happens basically, at 1-methyl donor, this slide I have not shown. This 1-methyl, this one carbon is being donated here and methionine is getting converted into basically succinyl CoA, that is why it is called a glucogenic amino acid. Now, this slide is new, this in one slide is important. 3 or 4 things you will know from here, this slide. One is this formation of active methionine, 1 carbon metabolism, and also formation of conversion of cysteine from methionine synthesis, cysteine synthesis from methionine. So, methyl bond is here in this ester form. So, that is to be released. So, this has to be some energy is to be given. So, S-adenosyl methionine will be formed and that is done by the methionine adenosyl transferase. This is the one important step I must tell you. So, this will form S-adenosyl methionine and that is the most active form. There is an active form of methionine and that is why I am telling you it is giving energy. And here you see the bond is here, S is here and S-adenosyl is here. Now, it is getting converted into adenosyl, homocysteine, and from adenosyl homocysteine is produced. From homocysteine to cysteine synthesis will be there in the next slide. So, homocysteine, what is happening? We require 1 more carbon to be attached and then this will form methionine. So, this 1 carbon methyl will be added with the help of vitamin B12. So, homocysteine methyl transferase will be there. So, N5 methyl tetrahydrofolate, 1 carbon will be required. It will be provided by the N5-methyl tetrahydrofolate through cobalamin to methylcobalamin, and this will be provided here along with the vitamin B12. So vitamin B12 deficiency is there, so this will not be formed. N5, N10 will not be formed, so it will remain trapped within the tetrahydrofolate. So vitamin B12 deficiency is there, tetrahydrofolate trapped at N5-methyl tetrahydrofolate and cannot transfer methyl group. So this is one question, methyl folate trap you read. So you will remember. So if there is a vitamin deficiency, then homocysteine to methionine formation will not be there. So again, vitamin B6 deficiency can cause this thing, vitamin B12. So B12 deficiency will cause defective formation from homocysteine to methionine. In the next stage, from serine you are having, you see the look at the structure of the serine. Serine is same thing. Only thing is that you know carbon, hydrogen and nitrogen, these three things, carbon, hydrogen, nitrogen are coming from serine. Only SH group is required from methionine, and that is provided by that homocysteine. It is a complicated pathway through which cysteine is formed. So, and from cysteine homoserine is formed. Now here you see this structure is important because it requires vitamin B6 to be active. Now, as a cofactor, if this cannot act, then cysteine cannot be formed. So if there is a cystathionine deficiency, beta deficiency, enzyme itself, or the vitamin B6 deficiency, or if there is a vitamin B12 deficiency, all will lead to homocystinuria. Homocystinuria is important in the cells because it causes cardiovascular and neurological disease. So those patients who are having cardiological disease, they are usually asked that what is the level of homocysteine in the blood. If the homocysteine level is blood, maybe it is due to vitamin B6 or B12 deficiency. Without knowing that thing, he will unnecessarily start treating the patients because the basic root cause of homocysteine formation is tetrahydrofolate acid, that is folic acid, vitamin B6 and B12. I know you are getting bored. This structure I have shown in the amino acid class, this cystathionine formation of serine from methionine, the same structure what I have shown here, this was taught in your amino acid class. And the final stage is propionyl CoA. Propionyl CoA, if you in your beta oxidation, you will see there will be even chain amino acid, there will be odd chain amino acid. Now, odd chain amino acid, even chain amino acid will be broken down finally to acetyl CoA. Odd chain amino acid will be converted into, at the end, one acetyl CoA, one propionyl CoA. Suppose 5 carbon atom will be there. Now 5 carbon atom cannot be broken into 2 acetyl CoA. So 1 2 unit, 1 3 unit. So propyl CoA is one important aspect in the sense that at the end of even chain fatty acid breakdown, the end product will be acetyl CoA and propyl CoA. Propyl CoA can also enter into the as an end product of beta oxidation of odd chain amino acid. So this is the structure what I have shown in short. This is available in the book, you read it. So while you are donating, there must be an acceptor for the methyl group, and this is the this chart, you remember, this is available. Some homocysteine, I have given you an example, that is methionine. Guanidinoacetic acid I have shown during creatine synthesis. Norepinephrine, methylated product is epinephrine, epinephrine methylated product is metadephrine. Like this, N-acetyl serotonin, methylated product is melatonin. Okay. So arginine metabolism, if you don't want to listen 2 minutes, then I have gone, only this structure was shown in the amino acid class. You see guanidino group is here. It contains, it is semi-essential. Why it is semi-essential I have told you, because it is required during the early part of life, during growth of the children and during recovery or from any illness where the demand of the protein is high. That's why it is called semi-essential or conditionally essential. So again, metabolic fat, if you look at this thing, arginine is a glucogenic amino acid. So urea cycle, if you remember, this arginine is getting converted into ornithine. So it contributes to ornithine formation. So breakdown product of arginine is ornithine. Glycine formation, I have shown this slide in the glycine metabolism. It participates in the kynurenine formation. Now this slide is new. This is arginine to nitric oxide. This is the summary of the breakdown products of arginine that I have also covered during your amino acid class. So now slide is new, only this slide. I am telling you all the previous, in the previous classes everything was covered, because I thought that if I do not take this class, I will cover that thing. Now this last part you note down only. From ornithine, from arginine, this polyamine synthesis is done. The three polyamines are there, that is putrescine, spermine and spermidine. This you wrote down. This is the only thing I want to tell you, and with this I am making an end, because I know this is a long class. I told you that I have to take class. I have wasted some time in showing you the class, otherwise the class would have ended at 1 hour time. So anyway, it is very difficult to remember all the points, all the cycles. You have to tell repeatedly. Even if you remember the cycles, question will not be asked straight cut from the cycle. But in a complicated way, just as I told you, vitamin B6 deficiency can cause pellagra-like symptom. Vitamin B6, B12 deficiency can cause that pellagra-like syndrome. That type of answer you have to give. Xanthoniuric aciduria is formed due to vitamin B6 deficiency. Explain why. This type of question you have to face. Okay.